everybody, welcome to our formal lecture video on titrations. So there's only about 10 slides to go through, and really this is just an introduction into what they are. So we're going to talk about exactly what a titration is. This setup you see in front of you is really fancy, but it is the most commonly used to track a titration. Now, a titration is going to be known as this process of taking the known concentration of something, and that's usually in the burette that you see on the left, and adding it to an unknown concentration of something else to figure out what the pH is at certain points. And we'll talk about what those certain points are. One of our key buzzwords is what we call the equivalence point. We will come to that here in a little bit, just like we have already kind of seen in our reading. Okay. From there, we can talk a little bit about, and I'm going to see... One of the things that we can also talk about is indicators. So a lot of times when we do titrations, we pour something into our unknown solution, the thing in the beaker at the bottom of the fancy burette looking thing, and we make that thing a certain, it'll do a certain reaction. So for instance, if we're trying to calculate what its pH is, we can use a dye that will actually change color at a certain designated pH equivalence point. So remember that the equivalence point is actually where the stoichiometric amount, so the number of moles of acid, will equal the number of moles of base present. So again, equivalence point means when the amount of the acid that you have added to base is equal in terms of molar amounts, not volumes, molar amounts, okay? We will talk in depth about that here in a bit. And this is our first look at a titration curve. So there are a couple of things that I do want to point out. On this titration curve, the axes on the right, let me see if I can get my little pointer dude up. The axis on the y is always going to be pH of the whole solution. And the axis on the bottom is going to be whatever we are adding. So if you remember from our first setup, I'm sorry, my handwriting, this is awful. If you remember from our first setup, we had our beaker, and we had this long, pointy device called a burette. Our burette had base, our beaker had acid, and we also had a pH meter in the actual acid. So what we were going to do then is we're going to slowly but surely pour drips of base into this solution one at a time. And as we add base to our acid, we're going to see our pH change. What we're looking for is this really steep part of the curve where the pH changes suddenly from either acidic, since we're adding to an acid, to basic, or from basic to acidic, depending on what we're adding to what. So in this case, since we're adding base to acid and we're tracking the pH of the acid to begin with, our initial pHs should all be in the acid range. Once we add enough base, we're going to see this curve steeply change, and eventually we're going to see the pHs become basic, and this is after the equivalence point. This is before the equivalence point. So we're going to see sort of semi-flat curves before and after the equivalence point. We're going to see this really sharp region in between. Now, because this is a strong acid and a strong base, this region is very, very sharp. This is the titration curve of a strong acid being titrated with a strong base. They are very characteristic, and we'll look here in a second at what a weak acid-base interaction is going to be. We're going to talk in depth about both types when we're doing calculations. That is our next big thing. So I'm going to film this video, and then I'm going to film a couple of different practice problem videos for you as well. Okay. Moving on, just to know, the before and after of the equivalence point, which is what we're just going to discuss a little bit further via the lecture, is that the pH is going to change very, very rapidly. What they're talking about is this super steep part of the curve before and after you hit the equivalence point. Now, before you even get close to the equivalence point, you see a pretty flat part of the curve, and after you see a pretty flat part of the curve. At the equivalence point, specifically, the number of moles of acid equals the number of moles of base. That's what an equivalence point is. The solution could have as many volume or as much volume as needed to get to this equivalence point. Don't worry about volume. We're going to account for that in our calculations. But for now, understand that mole amounts have to be equal. Okay? As more base gets added, then we see the second part of the steep part of the curve, and then we get up here and our pH levels are going to again flatten out. Okay? With a weak acid base, you can see what happened to the curve. It got squatter, right? Like, so it kind of got compressed on both sides. We see a little bit more of a steeper part of a weak acid. Remember, weak means doesn't completely dissociate. So in trying to get the acid to kind of break apart and become the base, we're having to put a lot more in to do it. And we're seeing that pH changes a little bit faster when it comes to a weak acid being titrated with a strong base. Now remember, this is a weak acid with a strong base. The inverse would also look very similar. But instead of seeing this particular graph, we might see a weak base has a really steep leveled off area with that equivalence point area in between, and then a very flat acid part. Remember, so if we flip this, we're talking this is the weak base. We're going to see a, again, steeper part of the curve here. We're still going to see a little equivalence region, and something my diagram doesn't really show you is that this should be compressed. And when I mean compressed, it means the whole thing probably needs to be down a little bit more. So remember, we see this like steeper region, then we see the little equivalence region, and we level off at low pHs for a strong acid versus a weak base. So again, remember that the thing, when you're looking at these graphs, in order to figure out what's being added to what, you always want to define on the left part of the graph, are the pHs very low? So are they strong acids? That means they're in like the 2 to 4 range. Is the pH kind of rapidly changing on this side? Is it a steeper curve? That means weak acid. And the inverse, so if we had really high values for pHs, we would see strong base is the thing we're adding to. And if we saw, say, a weak base, we'd probably see more of this. A strong base, we'd probably see more of this at the left side. And then on the right side, if we know that we are adding strong acid, we know we're going to see low pHs. If we're adding a weak acid, we're going to see that leveling. Okay, so it's just kind of how to interpret our titration curves. For now, when we're looking at the weak versus the strong, just understand that a weak acid takes a long time to get its leveled off curve, if you will. And what that means is you're going to have to add probably not necessarily more base, but you're going to see a lot better changes in the pH. Like you're going to see larger changes. This starts at about 2.5, and before the equivalence point, it's all the way up to 6.5. That's a 4-point range. That's huge on a logarithmic scale. So just kind of keep that in mind. Next thing to realize is the pH equivalence point is at about 7. The pH equivalence point is actually greater than a pH of 7, which is where you would expect a strong acid base to be. In our cases, it's above 8, and this is because this the conjugate base of this weak acid is also a weak base. So you're going to have to go back into chapter 16 to kind of remind yourself. The conjugate acid of a strong acid is a blank. The conjugate acid of a strong base versus the conjugate acid of a weak base. Remind yourself of some of those things. In this case, we're actually kind of inserting some knowledge here that we're going to talk a little bit more about if we ever get to see each other face to face again, on how to choose indicators for specific things. So with these titrations, we choose an indicator based on where we think the equivalence point will be. For this case, we could choose phenolphthalein, which turns bright pink when you add to it, at a pH above 7. So when you hit the equivalence point, you know you have those equivalent molar amounts, and you know that you have hit it because it turns pink. That's all these different indicators are used to do, is to tell you if you've gotten to the equivalence point or not. All right, just to kind of understand exactly what's going to happen in our next video on our just different stuff that we're going to go over as far as different types of calculations. We're going to start with what we know we're given. We're going to draw ice tables and kind of draw information from the ice tables, but maybe not actually use those ice tables. And then we're going to look for what's going to happen at equivalence. So there's a couple of different types of problems. The first is going to be, what is the pH before and after we hit the equivalence point? And the other is going to be, what exactly is going to happen at this equivalence point? So again, we're just talking about equivalence points and how we're going to move through some of these different videos. All right, so just to kind of give you an idea of what happens to the curves. The weaker the acid, right? Remember, we had this conversation about KAs. The weaker the acid, so the smaller the KA, the more compressed the curve ends up looking, right? So the initial part ends up being very steep versus look at what our strong acid does all the way down here. It has to use this huge jump to get to the equivalence point. Notice that as we move through these curves, the same amount of volume of NaOH is used to get to the equivalence point. And we'll talk about how that could possibly be the case here in a little bit. Okay, just kind of looking at some of those different indicators. Remember I said we use different indicators that react at different equivalence points. So for this particular for this particular base, remember it's a base because its initial pHs are above 7 and its final pHs are very low, so probably a weak base with a strong acid. Just make sure you understand how to read these curves. One of the things we're seeing is if we try to use phenolphthalein, which is the indicator we talked about before, the equivalence point is not in the range that the phenolphthalein indicator actually color changes. So using this indicator would be it would make no sense. We would not see a color change when we hit the equivalence point. In fact, we'd miss it completely. But if we use a different indicator, one that has a color change interval where our equivalence point lies, then we would actually see the color change. So this just goes to kind of give you an idea of how we choose the different indicators that we will choose. And we'll look at, I used to do a lab with this. I don't know what I'm going to do next, but we'll look at how to identify these things. Last topic for this video is just going to be polyprotic acids. So remember, poly means many, protic means protons. So many proton acids. And here we have an example of H3PO3. So what we see is for every proton that pops off, there is an equivalence point. And as we go from the first one to the second one to the third one, we're going to see that our equivalence points are going to be higher and higher as far as pH is concerned. And that's because as we remove protons, this thing becomes less acidic. Remove another proton, this thing becomes less acidic, and so on and so forth. All right, and that's where we're going to stop. I will start working on the problems next.